Thank you. I'm host. Respected chairpersons, my dear friends from India and France. Good afternoon, all of you again. So, first of all, let me thank the organizers for giving me the chance to stand here and present my works on Begomoviruses. So, I'll be presenting another 10 or 15 minutes. I'll be talking about the Begomoviruses and their something happened. The title is very small, but I made it very big but small. Oh, it's okay. So, Begomoviruses with the special reference to cucurbitaceous crops. I'll be like to talk and discuss about their diversity and how the variants are coming up one by one to infect vegetable crops, and also their management strategies, present strategies available, what all the future strategies we need to have in order to contain the spread as well as the control of the disease. Next slide. So here, first like to give an introduction about the vegetable crops. As you are aware, vegetables are the major source of vitamins, minerals, dietary fibers, antioxidants, etc. And in India, vegetables all put together, which is cultivated in around an area of 12 million hectares with a production of around 220 million tons. India is the second largest producer of vegetables next to China, and the annual losses due to these viral infections. As far as when we talk about the diseases of vegetables, I was wondering, our friend from Ooty did not talk about the virus disease in potato. Of course, they are concentrating on fungal and other bacterial diseases, but the virus diseases, no. The loss is varying from maybe 10 to 100, even 10%, because it totally depends upon the time of infection and the type of the viruses involved in causing the infections. So there are about more than 200 viruses infecting vegetable crops put together from different groups of viruses or the genera of viruses like Begomo, Pati, Taspo, Kukumo, Crini, Poliro, Tobamo, Como. Several viruses are infecting, and the problem now is the mixed infections, especially in vegetable crops, that make the thing very difficult to control the disease. And the reasons are the intensive cultivation. Throughout the year, for example, the villages which are very near to our campus, Tondamutur, throughout the year we will have tomato crop. So the inoculum, not only viruses, all pathogenic inoculum are always present throughout the year. So stage by stage, even single farmer, due to the price flexibility, the farmers used to grow or raise the crop in different months. So thereby continuously within his own field, the inoculum goes from one field to the next field likewise. So throughout the year we will be getting. So that intensive cultivation as well as continuous cropping are the major factors which are determining for the severe occurrence of the diseases, especially the virus as well as the Begomoviruses in general. And in regard to the virus history, our plant virus research was 100 years old, more than 100 years old. But viruses are very simple in composition, only the nucleic acid and coat protein. Maybe in few viruses will be having envelope. Very simple, but they are available in variety of shapes, and though it is very tiny, but most difficult to control as well as we experience corona. So for example, as far as the plant viruses are concerned, during 1939, there was only 89 viruses, total numbers infecting all put together vegetable, horticulture and agricultural crops. But slowly there was gradual increase in the number of viruses, viral species, and now more than 250 as on date, because after the submission of or dumping of data from the NGS, it has already crossed to 2250 species. So that is the status of the plant viruses as far as our cropping system is concerned. So these are all the major group. I have just mentioned few of the viruses. Even with the Begomo, not only tomato leaf curl New Delhi virus, which is one of the predominant virus in Tamil Nadu and also in India, but however, there are several variants from different regions within India that we will be looking that one. and also in okra, both yellow vein as well as Venetian leaf curl, chili leaf curl, several viruses in cassava mosaic viruses, and similarly in party papaya, ring spot, ziguni yellow mosaic, chili veinal mottle, then in cucumber virus, cucumber mosaic virus, which is one of the dangerous, and tabamo virus, cucumber green mottle mosaic virus, and TMV, tomato mosaic virus, as well as the TOSPO viruses, the necrosis symptom causing viruses like TSV. As morning we had no TSV on groundnut, cotton, groundnut button necrosis virus, capsicum chlorosis, watermelon button necrosis. They are very severe outbreak we are getting in India. Similarly, the polioviruses. These two groups are emerging now. But though we are talking about the begomoviruses are emerging, but already emerged, but new viruses are also emerging in that group one by one. So as I said, they are all in a variety of shapes. See here, flexuous rod, spherical, or pleomorphic, or bacilli type, or different types. But the begomoviruses are Gemini. They are all twin particles. They look like the bullets. So based on that only, they have coined the name. The geminate or the Gemini or the Begomoviridae comes under the family, and they are twin particles. Either they are monopartite or bipartite, with the approximate genome size is very simple, 2.7 kb only. But there are about 14 genera under this family, but the begomoviruses alone has about around 70% of the viruses coming under this begomovirus from the Begomoviridae, and associated with the satellite also. I think that Dr. Urbino going to talk about that satellite viruses tomorrow, satellite molecules, and both the alpha as well as beta. Delta is also there, but we predominantly getting only alpha and beta satellites. And similarly, they are infecting, there are about 243 begomovirus species infecting only vegetable crops among the horticultural crops. And Bemisia tabaci whitefly transmits the virus in a persistent, circulative, but not propagative manner. So pathogen detection, we need to know what is that. Nowadays, no confusing whether it is due to deficiency or due to some other fungal or bacterial pathogens, because necrosis is also one of the symptoms caused by the viral diseases. So we have several methods. Of course, here for the pathogen detection, the cultivation may not be possible for our viruses, but other serological as well as the molecular methods, either protein-based or the nucleic acid based methods, or the sequencing as well as the biosensors and the non-invasive detection methods are all commonly used for the detection of the viral diseases in the horticultural ecosystem. So I'll be talking about the cucurbitaceous begomoviruses especially. So we have 7 agroclimatic regions in Tamil Nadu. Throughout all the zones, we will be cultivating cucurbitaceous crops, except a few areas like high rainfall area and also some of the other hilly areas also we have the chow chow and all, or chayote. But however, in all the places, we will be recording the begomoviruses and the characterization. So you see first the symptoms. When you talk about, variety of symptoms are producing. So you can just for to know the crop, familiarize with the crop, I have included the fruits also. So the bitter gourd, they produce different symptoms. The leaf curl, yellow mosaic or ordinary mosaic or mosaic mottling, and similarly on pumpkin also you can see kind of chlorotic spots or chlorosis, as well as the mosaic mottling look like yellow mosaic. And on the ridge gourd also you can see yellow mosaic, and here yellow mosaic on ridge gourd, leaf curl, puckering also could be seen sometimes. Then on the ash gourd we will be getting mosaic, yellow mosaic as well as the yellow mosaic. And on the bottle gourd also you will be getting mosaic, a typical mosaic mottling, and the filiformity of the leaves, or sometimes they become narrow, leaves become narrow. And on the ivy gourd also you will be getting yellow mosaic, and snake gourd, typical characteristic mosaic mottling can be seen. And on cucumber, there also yellow mosaic will be common symptoms. And muskmelon also, yellow mosaic is the predominant, and the leaf malformation or deformation of the fruits also could be seen in majority of the crops. And similarly in chayote also, typical mosaic mottling as well as the puckering of the leaves are the additional symptoms. This is all severe outbreak with yellowing or yellow predominancy on the leaf surfaces. So normally these begomoviruses are characterized, diagnosis is easy, but they are characterized by a special method, by the amplification of the genome by RCA, rolling circle amplification. It is a simple isothermal reaction by using the special enzyme called 529 that will able to produce concatenameric DNA. So from that, by restricting those sequences, we will be getting the 2.7 kb genome of this viral sequence. So that is highly useful for the diagnosis as well as the comparison of the viruses. So this is one of the based on general group. They formed the viruses, the begomoviruses which are infecting the vegetable crops, mixed infection, are only on solanaceous crops, and solanaceous and malvaceous crops, solanaceous, cucurbitaceous vegetable crops, and legumoviruses and all. They have group, but however, they are either monopartite or bipartite, and also the involvement of the satellite, either alpha satellite as well as beta satellites. Peanut butter necrosis virus, that total plant will be collapsed. In Tamil Nadu, 73% of the tomatoes are produced from these four districts: Dharmaburi, Krishnagiri, Salem and Dindigul. And the symptoms are very starting from leaf curl, but we don't get yellowing symptom here. Only leaf curl, stunting, malformation of the leaves, even sometimes fruits also. But we'll be getting some of the weeds also get infected with the same viruses. In the ecosystem of tomato, we have collected several weed samples, and that also turned positive for the viruses which are acting as the
5 more minutes.
Okay, sir. So collateral host for the viruses. So here, see, at that time, when I was starting my research on viruses, maybe 15 years back, we could get only tomato leaf curl New Delhi virus. But nowadays, even in Tamil Nadu itself, majority of the genomes are aligning with the Bangalore virus only. But in this particularly in these districts, we have randomly collected and analyzed, we are getting different viruses, not only New Delhi, and also Palampur, Bangalore, Gujarat, Kerala, and also satellite also from different categories, Bangalore, Gujarat, and also Multan. Cotton leaf curl Multan alpha satellite was also present. That's what I asked that question. The cotton leaf curl virus is not present in Tamil Nadu, but the satellite is present, alpha satellite. And similarly, the beta satellite of tomato leaf curl New Delhi virus is also obtained in the tomato sample. So this is the scenario. So all are within these groups, one or two as a mixed infections, in some cases single infection, or in some cases with other viruses, not begomoviruses, with other viruses like CMV sometimes, or cucumber mosaic virus, or with peanut butter necrosis virus, also occurring as a combined infection. That is a major menace nowadays, the combined infection. So this is the phylogenetic tree constructed using the DNA A of tomato leaf curl, New Delhi virus, Palampur virus, Bangalore virus, as well as the Gujarat virus. They are all formed different clades, separate clusters are formed with them. And similarly, see, DNA B also, they are forming a distinct group, and the alpha satellite also, they are aligning with the concerned group of alpha satellite, whether they are tomato leaf curl or Bangalore, New Delhi or Bangalore, like that. So this is the scenario again. So this is the Gujarat virus as well as the Gujarat beta satellite also we have characterized. And recombination analysis also we did, and some of the viruses between the isolates from one crop to the another crop, like tomato with the cucurbitaceous isolate, they have recombinations either in the DNA A or in the DNA B. A small portion were identified in the recombination analysis also. And with this background, we have developed the infectious clones for the cucurbitaceous begoma, one of the cucurbitaceous tomato leaf curl New Delhi virus, and we have developed the agro-infectious constructs, and that agro clones were inoculated on different, in order to know the host range, on different cucurbitaceous grass, including the tomato. We could get nearly 10% infection in some of the cases, like ash gourd and also squash. And on tomato also we could able to get more than 90% of the infection by this infectious clone. So and also on wheat plants also we could able to, but when you add, in addition to DNA A and DNA B, when we include beta satellite, the infection rate was found to be high. Really low when they are inoculated without the beta satellite of these viruses. See, these are all the symptom pattern on the inoculated, agro-inoculated cucurbitaceous, various cucurbitaceous crop plants, and the titer was also determined. Titer was also found to be very high in the infected plants. And similarly, in the beans also, we could get both in the French bean as well as in the field beans, we could get this Begomovirus, Dolichos yellow mosaic virus, and that was also aligning with some Washington Dolichos yellow mosaic virus and other Indian strains or isolates from Kanpur and all. That is also distinct. And there are similarly now in the brinjal also there is a complex of Begomoviruses are infecting with the predominant occurrence of tomato leaf curl New Delhi virus and also the Bangalore virus. And chili also. In chili, mostly we will be getting chili leaf curl virus, and in addition to that, we will be getting the tomato leaf curl New Delhi virus also on chilies. And also in Bendi also both okra yellow vein mosaic virus as well as the Venetian leaf curl also we will be getting nowadays. Of course, nowadays that yellow vein is reduced incidence, but however the Venetian leaf curl was found to be severe. And seed transmission of these Begomoviruses on cucurbitaceous crops was also proved, and that resulted in a transmission of around 12%. So for example, in one of the hybrid, it was found to be 12% seed transmission, and the another one, it was found to be 5%. This is one of the next problem, for the outbreak and the severity of the diseases. And similarly, other cucurbitaceous ridge gourd also, we could able to get 18% seed transmission in one of the lot, but however, in one of the other hybrid, we could not get any seed transmission. That was proved. And the major next problem is the mixed infection. Within the cucurbitaceous crops, see here, these are all the different combinations of the viruses get in the natural field conditions, but in all the combinations, we will be getting the Begomo. So without Begomo, there is no combined infection as far as the cucurbitaceous are concerned. So that is the scenarios. Then we can imagine how much the Begomo incidence is occurring under the field conditions. Similarly, in the whitefly also, like that of the virus, Begomovirus, whitefly also there are different variants, and there are called as biotypes, cryptic species, that also we could able to identify. So for example, in the cucurbitaceous ecosystem, that correlated with the incidence also. For example, in cucurbitaceous ecosystem, mostly we will be getting that Asia 2-5 group of cryptic species of whiteflies, Bemisia tabaci, and wherever we are getting this 2-5, Asia 2-5, there the incidence was found to be high when compared to the other Asia 1 or the other cryptic species. So this is the grouping, and here also you can see this is the 2-5. It's a very big range when compared to the other types. And in cassava ecosystem, we could get, especially in Salem and the Namakkal and other major growing areas, we will be getting only one group, Asia 1-5, not other group. And similarly, that's and regarding the management, we have good options, integrated management, starting from the resistant varieties. Dr. Andy Kumar was talking about the microbiome, the biologicals, the biological control, barrier crop, and other cultural practices are there. That also we can follow for the management. And the recent technologies like genome editing or double-stranded RNA application, exogenous as a non-transgenic approach, that is also we are working. We have developed the dsRNA strategy for tomato leaf curl New Delhi virus, and also we have come out with the another one virus, which is also Begomo, but infecting the legumes. For that also we have developed the dsRNA formulation with nano-based formulation also we have come up, and that found to be highly effective under the field conditions. But however, we need to give up approval and other field studies we need to conduct. So summary regarding my presentation, as we see from the presentation that the frequent presence of or the occurrence of mixed infection is the major problem as far as the cucurbitaceous ecosystem is concerned, even other vegetables also. So the recombination between strains and our species playing a critical role, and vector pressure and their environmental factors as well as their cryptic species also play a major role, and the weed host, the reservoir plants available in the concerned vegetable ecosystem, that also play a major role for the survival and the spread of the disease. And the future strategies, though we have quarantine, but there is no strict quarantine, I can say, in the closed wall, because how this Philippines virus has come into India. Likewise, there are several viruses which were introduced from other countries like Pakistan and other neighboring Bangladesh and all. That also we need to take care. And similarly, the climate resilient crops adopting new disease pattern, and the AI should be exploited for diagnosis, disease forewarning, and spot delivery of inputs, smart surveillance and modeling and all. They were talking from the morning. Then policy support is very, very important, and the circular economy. And finally, by combining artificial intelligence, genetic engineering, nanotechnology, biocontrol, and climate smart practices, modern agriculture can achieve resilient, high yielding, and sustainable vegetable production systems in the globe. These are all some of the papers we have published from good journals. And I need to acknowledge my sponsors, DBT, SERB, ANRF, as well as Science and Technology, Department of Science and Technology of Government of India, and the USAID, Virginia Tech, Washington State University, and also DSMZ for supplying especially for the diagnostic purpose, they used to supply antibodies. These are all my team, my boss at EVS, Nadu Rayapati, when I was doing my postdoc. Then Muniyappan, he was the director of IPM CRSP, and Dr. Nagendran, scientist, tomorrow he will be attending, and my students as well as my research fellows from our laboratory. Thank you very much for your patience. Thank you.
Long live India and France friendship.